billions of years, evolution has been making these tiny little factories. I'm going to give you two examples. One, a seed that becomes a plant. The other, the fundamental unit of life, is a cell. We're made of them. Is it possible to go into something as large as a seed or as small as an individual bacteria cell and edit it in a way that would unlock opportunity for humanity? Right now, humanity makes trillions of dollars of stuff, medicine, chemicals, fuel, in very 20th century industrial processes. What if we could use biology to help us make humanity's future? That's what we're going to talk about today.
There is so much we can do with these miraculous solar-powered, carbon-negative, self-assembling machines that we call plants.
Our mission is to enable more and more of the world's materials to be made with biology.
I'm Astro Teller, your host and captain of Moonshots, and this is the Moonshot Podcast. To kick things off, we're going to look at the future of what's on your plate. Joining us today, we have Brad Zamft, the CEO of ex-spinout Heritable Agriculture. Welcome back, Brad.
Thank you. It is amazing to be back. What a great, magical place.
We're going to spend a lot of time getting into the details of what it means to program plants and how it could change the world. But on our way into that, does the world really need better plants? What's wrong with the plants we already have?
So the agricultural industry has done a miraculous job. $3 trillion industry, 10 billion tons of biomass that we call food is created every year that feeds an ever-growing population. And a population that's quality of life is increasing too. So not only is the number increasing, but the amount of food and the intensity of that food production increases. And the conventional way of improving crops has enabled that. So we have a lot to thank the farmers and the agricultural system. But what got us here won't get us there. As population increases, as people rise up in economic status, we have to keep the innovation and the pace up. We can't let the curve flatten.
Since you talked about coming into this building, remind us how you met X in the first place. What did it feel like when you started here? Because you weren't a deep plant nerd then.
I was on my way to becoming a deep plant nerd. My career trajectory is complicated with lots of twists and turns, which are always the best careers to have. And I got deep into synthetic biology in graduate school and a postdoc that I did. Just absolutely fascinated with the idea of uncovering the code of life, really understanding the inner workings of a cell. And then I went from postdoc to a fellowship in the United States Department of Energy in the Advanced Research Projects Agency. And there I helped, I assisted building grant programs on biofuels and on robots that would go around the field and measure plants. And there it kind of struck me that, I had been spending all of these years figuring out how to program microbes, but the entities that take up 40% of the non-glacial landmass, that use most of the water, that provide all of the food, those biological systems are really underdeveloped in terms of our ability to optimize them. So that kind of sent me on a long journey. I went again through a bunch of twists and turns in the career, but the one that stuck was the one that had been there since my entry into the Department of Energy.
Do you remember the first time you tried to tell someone else at X that you wanted to program plants and why you wanted to program plants?
Yes, I remember it, although it was a process. There wasn't a singular moment. I knew the idea was good. I have absolute conviction that we need to have a step change in the way we optimize our agricultural system. That took me a couple months to figure, I think I want to go for it.
So let's pretend somehow we have a black box and we can ask the black box for any kind of seed to make a plant. It's like a genie for making plants. What would you ask it for? Like bigger strawberries? What would change the world?
First, the number one trait that affects agriculture's impact on the planet is yield, resource efficiency. Can you grow more on the same piece of land with less inputs? Everything follows from there. We will have less water use, less nitrogen use, being able to feed the planet better if you can increase that efficiency.
Just to make sure we're tracking. So that's like for a corn stalk that goes up, if there was 2% more juicy kernels at the top of that, that counts as more yield.
Right. Keeping the amount of inputs, water, fertilizer the same or dropping it. That is what is going to move our society towards more sustainable outcomes.
Okay, so yield.
That's the number one thing.
What's number two?
Unanimously, when I talk to folks in the industry, each one of them lists a pest that they are deathly afraid of. We have, in the words of the head of R&D for one of these major companies, they have a COVID every year, somewhere on the planet in some crop. So the generation of new diseases is decimating our food production system in certain cases. The ability to dial in and say, we see this pest coming, we're going to generate a resistance to that, that is a holy grail. But so is flavor, color, shape, disease resistance. So these are the kind of things that as we improve our sophistication and our ability to improve plants, it unlocks kind of all of these different things.
Would drought resistance also be in that list?
Drought resistance, heat resistance. Yeah, all of that is are major problems facing the industry. And everybody's trying to solve them. Can we solve them better, faster, cheaper?
You have this vision that being able to reprogram plants to get at plants that have some of these characteristics in a much better way would be phenomenally good for the world and a phenomenal business. But can you take a pause and just, in the simplest words you can, describe what you mean by reprogramming a plant?
We've been programming plants for 9,000 years. Our ancestors spotted this weedy-looking thing with a couple kernels on it called teosinte. And they found the best one, the biggest one, the one with the most kernels, and they propagated that. That's called breeding. And we've been doing that for millennia. You're just selecting the best plant from its emergent properties, from its physical properties. But what you're actually doing is you're finding the best genome in that environment.
You're biasing a random walk in the genome space of the plants.
That's right. So we've been doing that. We've been programming plants for all of modern agriculture. Recently, we've got computers and we've figured out that if you look at the genome, you read the genome, you do DNA sequencing, you can actually build models that predict what the plant will look like before you grow the plant. Or you'll predict what the outcome of two matings, what that child will look like. That's called breeding.
So we ask a computer to guess if we took some DNA from this plant and some DNA from that plant, and they're both, let's say, corn plants, and we mush them together, what will the baby corn plant, the new corn plant, actually do when it grows up?
That's right. We used to do phenotypic prediction. A phenotype is that emergent physical property of the plant. Now we've moved towards genotypic prediction, using the information that we can gather from the genome to predict what the plant is going to look like or what the children of a cross will look like.
If you imagine the DNA sequence that describes in this double helix inside that seed what the seed should do when it grows up, that's its program. Mm-hmm. How hard could that be to go in and just make some changes to that to make it better? Shouldn't be too bad, right? We're good at programming.
Yeah, a genome is a program. In it is the plans for how the plant will, or the organism will, operate in the environment. That program came through evolution. It's being coded randomly around the world with little sharing in between geographies for 4 billion years. But if you want to do better than that, if you don't want to take 4 billion years, you have to kind of disentangle that code. So how would you disentangle that code? What we've done recently is we can actually read the lines of the code. We can read the DNA. It is the plans that get passed down from generation to generation. But it says almost nothing about how the plant is experiencing the world at that moment.
Your discovery, which is now, I think, what Heritable Agriculture, your company, is doing that graduated from X, about watching the RNA inside the cells and how they're functioning.
So what's wonderful about DNA, it's inherited. It is the only modality in biology that doesn't reflect the current state of the plant. But everything else, RNA, metabolites, epigenetics, proteins, these are moving in real time, responding in real time to the environment, to the specific tissue that they're in, under drought, for example. So you can gain much more information by peering into those other pieces of molecular information. So that was one of the major unlocks that made what is Heritable today.
So now you have to start growing some plants. You have to find out if you're kidding yourself or not.
Right.
What were those first experiments like, and how did you get up and running here at X?
We started working with partners to identify ways to grow those plants and extract the RNA and DNA information to be able to build those models. We started growing model organisms, very small things that grew fast here at X. So we sort of took a portfolio approach.
Model organism, unpack that.
There's a plant called Arabidopsis thaliana. It's anybody that works in plant biology will roll their eyes when they hear it. It has no commercial value, but it grows fast. Over the decades, we've learned a lot about it. It's the thing that scientists use, very well characterized organism. It also has a very easy way to molecularly disrupt it. So we worked on that. We did that in parallel with working on the crops of agronomic value, but it allowed us to iterate really, really fast.
So what did you do? Did you try to turn it white, make it have more leaves, grow twice as fast?
You nailed it. We tried to change the number of leaves, and we did. We grew a lot of different Arabidopsis plants, genetically different Arabidopsis plants. We extracted their DNA, RNA. We built the model, and the model output which genes are associated with a number of leaves in the plant. We chose that trait because there was a lot of literature about it, and we could benchmark the models before even doing any experiments. As soon as the model had an output, we could look at the output and say, are we discovering genes that have been discovered before? And we did. Then we went ahead and commissioned some gene editing to be done. So very simple stuff. You just break the gene, or maybe you hit one of the parts of the DNA that changes how much that gene is being made. And you see if what the model said was going to have an effect had an effect, and it did. So that was very exciting early results. We did the same thing in corn. Now, corn is the number one most valuable crop in the United States. It underlies most of our food system. Flowering time, which is related to leaf number, is been studied for 40 years. And we went ahead and built our models, ran our trial, and in 18 months, we discovered new flowering time genes.
Tell us a little bit about, like, where did you grow them? Like,
Yeah.
This is a very high-tech building. Like, where was the farm?
That's what's one of the amazing things about working at X. So there had been a project that was winding down. They needed a very sophisticated growth chamber to run their project. And we had this big rapid prototyping room. It was empty, and we went ahead and we put hundreds of plants in there, all of genetically distinct plants. And we watched them as their leaves developed as they flowered.
So you've been working on this for maybe a year and a half. We have some good early evidence that there's something important here. Now it's time to get out of this building and start growing more out in, like, the real world. And then COVID hits. Tell me what happens.
Yeah, moving from the controlled environment to out in the real world, that's a whole other level of complexity, but also validation and an impact. So yeah, we ran some trials in Illinois. It was a couple of months after the lockdown. We had already been growing the plants, but we had to do the sampling, and the plants don't wait. We flew to Illinois and worked with this company to train their people to do the RNA kind of sampling, the tissue sampling for RNA. A few years later, we decided we wanted to investigate what was going on below ground. Everybody focuses on corn yield. They look at the things that are happening above, but there's this universe that's happening in the soil.
You mean the root system for the plants?
The root system. So let's rerun the trial that we'd already done, but let's dig up the roots. That's good in theory. What we ended up doing is hours in the sweltering Wisconsin heat of summer. It's hard to dig up a corn root. Mud sticks to it, so you have to have an entire operation about how to spray those off. And remember, the only modality that is not changing over time is the DNA. So time matters. If you're going to go measure the RNA in those roots, you have to do it minutes after you've dug them up. You can't go because the RNA will have changed. So we had to race. We would dig it. We'd pull it out. We would run through the field to the sampling station and immediately do the sample, freeze it in liquid nitrogen, and we had to do that thousands of times over.
You did start growing stuff on the roof of our building too, didn't you?
You were renovating the bushes on the parking lot. And we said, hold up, can we grow some corn there? And when the corn got big enough, we brought chairs out, and then I would have my one-on-ones inside a cornfield on the roof in the parking lot of X.
So take us forward now to graduation. You went from being a project at X to being a startup outside of X, Heritable Agriculture. Several of the people who were here with you moved with you to be part of that. Tell us a little bit about that.
It's very different. There's nothing that instills more discipline and more rigor than pitching 100 investors and getting 90 no's or whatever it is. I'm not disclosing our numbers. It was definitely a hard transition, but we are doing exceedingly well and showing the industry something new.
What's one or two things you feel like you purposefully brought with you from the Moonshot factory and you want to make sure that it's still alive and well at Heritable?
There's a lot of things that we took, but the one that I'm a broken record on internally is about the culture. The culture is tantamount. This is not a 9-to-5 job. We're trying to change the world here. So if you're going to ask people to show up to a job that is not a job, it's a passion, it's not a job, you have to create an environment where they feel valued, cherished, and passionate. You have to value the people, not the employees. They have to know that this is an investment in them as much as they are investing in the company.
Paint a picture for us. Heritable Agriculture is massively successful. It's 10 years from now. How are you bragging on yourself?
I mean, wow. I want to see an improved plant in the ground because of us. I'll start there. Something that improves the nutrition of the global food system, that when that day, I dream about that day. It takes a long time to grow plants. And it'll be a few years before the first improved plant is providing value for the industry. That's what I dream of. I'm just going to be very, very proud if we've made a dent in the efficiency of the agricultural system, if we've changed the lives of some consumer, some grower. That's why I'm here.
Right on. I would love it if, at least partly because of your company, that we're not just going to have a plant that everyone thinks is the good version of yams or the good version of potatoes or whatever that is, that at least going forward, we will design plants for the environments they're going into.
I'll take that further. The vast majority of the investment in agriculture, vast majority, goes into 2 crops and 1 trait, yield in corn and soy. We need a future where that level of sophistication, that level of improvement is happening for all fruits and vegetables, for our forests. Plants not only provide food, They don't only provide calories, they provide nutrition, they provide fiber. There is a world where they can provide fuel. They make this miraculous, long-lived, functional material called wood. So there is so much we can do with these miraculous solar-powered, carbon-negative, self-assembling machines that we call plants. The current agriculture system is just the beginning.
Brad, thanks so much for doing this with us, and we're really looking forward to seeing how heritable agriculture grows up.
This has been a blast. Thank you for having me back. It's so great.
Thanks. Brad just talked to us about the opportunity to help humanity by literally programming plants. Now, he's looking at starting from plants and going out into the world. But what if we went inwards instead? What if we went all the way down, much smaller than a seed, all the way down to the size of a bacteria? And in that tiny one-cell organism, we saw it as a potential factory for making things for humanity. If only we could learn to program life itself to make things for us. Can we harness biology in that way? Relly Brandman, a computational biologist and project lead for A-Life, is here to talk to me about it. Welcome, Relly. Thanks for doing this with me.
Thanks, Astro. Happy to be here.
You know, for the last, 5-ish years, when I've been on stage and asked for some crazy prediction about the future, the thing I've said more often than anything else is that I believe that the 21st century will be remembered as much for biology moving from a science to an engineering discipline as for artificial intelligence. And seeing what A-Life has been building and the progress that you've been making is what's mostly made me a believer that that's true. And so tell us, what does A-Life do?
A-Life is X's moonshot to unlock the manufacturing power of biology. Our mission is to enable more and more of the world's materials to be made with biology.
What does that mean to manufacture things using biology?
I'm guessing many listeners have enjoyed a cold glass of beer or a glass of wine. Beer and wine are made from tiny, tiny, tiny single-celled organisms called yeast. And they just naturally make the ingredients in wine and beer all day long. And humans have been enjoying that for a very long time. Fast forward a couple decades ago, scientists came up with very advanced tools in the biology lab that let you go into life, like yeast and like bacteria, and start to edit the DNA. And they can do that now with such precision that you can go into those same yeast cells that are making ethanol for beer or for wine, and instead have them make lots of different materials. Things like food, polymers, fuels, textiles for clothing, building materials, vitamins, medicines. Many people probably use biomanufactured materials all the time and they don't know it. As an example, have you ever washed your clothes in cold water?
For sure.
The reason that is possible is because there are enzymes put into your laundry detergent that can break down stains really easily, including at cold temperatures.
Is it like the spot remover have stuff that's made with biology too?
The spot remover, the detergent, it just removes stains, again, without the need for harsh chemicals or high temperatures. And so that's just one of the many very innovative materials that you can only make with biology.
Thinking about cells as these tiny factories for making stuff, if we zoom in inside the cell, there's so much going on in the simplest terms possible. Can you help us understand how that factory works? How could it come to make a super complex protein that it pushes outside of itself somehow?
Cells have two main jobs when they are making materials. One is to grow and one is to make the material. And the way they do that is they're going to take in sugar and nutrients from their environment. They're going to turn that sugar into energy, and then they're going to have all sorts of other biosynthetic pathways that they do, either to grow or to make the material that we care about.
Is the job of the cell to make stuff, or is it more like a side effect, like a burp that we have, where humans and cows aren't made to burp? That's just a side effect of us being, but you could make humans burp more by maybe giving them a carbonated drink. And so that's why when they make something that we want them to make, they kind of push it out through their membrane because they're like, oh, this is garbage. They think it's garbage, even though it's what we want.
That's right. They'll just make that naturally as a side product all day long while they're happily growing inside a fermentation tank.
So you described how for thousands of years, humanity has been asking biology to make something for us, the alcohol that goes into our beer and wine. You've said that over the last couple of decades, humanity has learned how to start reprogramming these bacteria by changing their DNA so that they make other things for us. So what do we need A-life for? I mean, if the world's already off and running, what's A-life?
The process of making materials with biology today is slow and expensive. That's because scientists know how to go in and adjust the DNA of cells, but they don't know what's going to happen after they make those changes. With A-Life, we're shifting that process from trial and error to predictive design. That's going to enable more and more of the world's materials to be made with biology.
I just want to drill in. Is it the actual programming of the cell that's expensive, or is it that you don't know what will happen after you change its genetic code in some way?
So as an example, let's say you want to make a medicine, right? You've gone through all this discovery, you went through the clinical trials, you have something amazing, and you're ready to start making it. Well, that starts another very hard problem. What you have to do is you have to start genetically engineering that cell. But cells are so complex that it's really hard to know what genes in the cell do you want to adjust and why and how. So scientists read papers, they try things. A lot of approaches look like random trial and error. Then once they do that, and that's usually done in very small volumes at lab scale, then they need to go from the lab scale all the way into a large fermentation tank. And when I say large, I mean large. It's the volume of something like a swimming pool. Very different environment for a cell than those tiny volumes. Then they have to go through the process again, where they start fiddling with the different fermentation parameters, like what temperature should I put it, what pressure, what pH, is it getting enough oxygen, how much glucose do I feed it? They treat those as separate optimization things, and often you'll get these, again, very late-stage failures that are very expensive. That's how things are done today. With A-Life, what we do is we think of that from the bench scale, the discoveries you need to make there, all the way into the manufacturing scale. We consider that one system, and we help optimize that as one system.
If I have a medicine and I don't want to go through all that, can't I just go get a machine that makes my medicine? Like, what if I didn't want to do all that biology stuff? I want to 3D print it or something. Like, what's the alternative?
So the alternative is chemical synthesis, but there are many molecules that are so complicated that you can't make them with chemical synthesis. You can only do that with the tiny factories inside of cells. And in fact, many, many medicines are like that. Protein medicines are like that. Antibody drugs, which a lot of cancer therapies are like that. In fact, as innovation grows in pharmaceuticals, over time, more and more things you can only make with biology.
Would it be fair to say that if you know how to reprogram a cell to get it to do things for you, but you don't know what will happen when you reprogram the cell, the whole thing is sort of a black art? And what A-Life is doing is moving that whole industry from being a black art to being an engineering discipline by making it explicit what you'll get when you reprogram the cell in different ways.
Yes, that's right. When A-Life comes in, because we're using artificial intelligence models and other things that help us create a map of the cell, we can significantly reduce the amount of experiments that scientists need to do in order, for example, to make a medicine.
Let's dive into, you have this software system that you've built that answers this previously unanswerable question, which has unlocked this whole industry now. Mm-hmm. How did you make that? In particular, if you have a huge amount of data, like we have text on the web, you can just throw it at machine learning and get a large language model. But that kind of huge data doesn't exist in your world. Experts have some sense of how cell biology works, but they mostly don't actually really understand it. And so you can't just write down the rules from the experts. How did you marry this sort of work of the experts and their partial understanding with the little bits and scraps of data you could get your hands on?
Biology data looks so different than internet data. In internet data, you typically have huge amounts of data, and that data is all one type, like all video data or all text data. That is not at all what biology data looks like. In biology, you typically have very small data sets, and those data sets are very heterogeneous. So you need machine learning algorithms that are going to work in that new reality. And we understood that. We understood we had to start from the bottom up. And so the history of our team is we actually started by encoding what scientists do know about cells. We're just scratching the surface of understanding those things, but a few things are known. And so we started by coding that into a computer program. In our lab, we grow bacteria and we grow cells to learn how they work. And very specifically, we're learning how they work, not so a human can understand how they work, but so machines, so artificial intelligent models can learn how cells work. There's 3 major activities that we do in our lab. One, you're going to see scientists on the bench. Those are the scientists that are going into the DNA of the cell and teaching that DNA how to make the materials. 2 is we grow up the cells. Lots of different ways we grow up the cells. Some is in tiny volumes. You'll see these 96 well plates. 2 is in bigger volumes. You'll see flasks that are about this big. And finally, we have fermentation tanks that are about 2 liters big that sit on the bench. So that's the second thing we do, right? So we edit the DNA and then we grow up the cells. The last thing we do is after we grow up the cells and we do put them in different conditions, we need to understand what is happening inside those cells. How can we take a cell signature snapshot? Right? Cells are so complex. There's millions of different components in there, millions of proteins, lipids, small molecules, metabolites. How do you take this biological goo and actually know what is in there? That's the kind of data, right? If you want to give data to an AI model, you need to know what is in the cell and how much of that thing. So that's where we have our analytical chemistry equipment. So for example, HPLCs or mass specs that can take that biological goo and spread it out and separate it into individual components and then figure out how much does each of those components weigh. And from that weight, you know what it was.
Would it be fair to say that kind of what A-Life has done is make a virtual cell?
Yes.
And so if A-Life has made a virtual cell, then instead of having to try cells in the real world, you can try cells in the computer.
Yes. We are modeling how cells work, but not just a cell in a vacuum. We are modeling how a cell works in its environment. So both the genes inside the cell and the environment outside of the cell. Then what happens is because we're modeling that, we can run millions of potential experiments where you change the genes inside the cell, or you change what's outside of the cell, the sugar, the food, the nutrients, the pressure in the fermentation tank, the temperature in the fermentation tank. We can optimize all of those things as one system and then guide the experimental scientists in the lab and say, these are the most high priority experiments to start with.
When you have this kind of virtual cell, this model of the simulation of life, you're imagining changing its DNA, or you change the pH in which it's sitting, or you change the concentration of sugar in which it's swimming. Your system makes a prediction about what's going to happen. And then let's say you're slightly wrong. How do you do the feedback on that, where the data that you get through the process you just described makes the system better?
We work in what are called design, build, test, learn cycles, where you make a decision for what experiment you want to run. That's the design. Then you build and test it. That's what you go in the lab and you make the changes that you decided to make. And then you learn. You see what happens after you make those changes, and you use that information to update your next design cycle. So today, before A-Life, right, then design looks like scientists reading papers or trying random things. Learn looks like scientists analyzing data. With A-Life, the design and learn now become AI guided. You can run millions of experiments for the design cycle, then pick the 10, 20, 30 highest priority experiments to go into build and test, actually go into the lab and try it. Then when the data comes back, you feed it back into your AI model and do it again. So when your design, build, test, learn cycles are AI guided, you need significantly fewer experiments to get much better results.
What are the first couple of products that customers, actual businesses around the world will experience from A-Life?
We work with biomanufacturers. Biomanufacturers are making ingredients with tiny manufacturing plants like yeast or bacteria or algae. So our customers are the world's experts in making materials with biology, and they already have in-house teams that are able to change the DNA of a cell and change the environment of a cell, and they have fermentation tanks to make materials. When they start working with A-Life, what we do is we add artificial intelligence into those workflows. We help simulate millions of different potential experiments that those scientists could be doing. We prioritize the most important experiments, and we make recommendations for them, the 10, 20, 30 most important things they should be doing in their lab next.
How about the automation in the labs,
Yes.
sort of building sort of to the point where you can be doing 10 or 100 times as much a year from now without having to have 10 or 100 times as many people?
Up until now, we've been doing a lot of exploring in the lab. And what we most wanted to know is what kind of data about cells will be truly unlocking for model performance. That's a breakthrough that happened recently. What that breakthrough means is that, of course, we're going to continue exploring, but we've discovered enough that we're ready to start scaling up. We're ready to move from A-Life scientists pipetting into a 96-well plate to robotic systems that can do many plates at once and significantly scale up the data we're collecting.
Now that you've made a virtual cell, do you think as like a side effect of A-Life that people might actually start doing other things with your virtual cell besides trying to reprogram the cells to make things? Like, is it possible that it'll become a platform for learning about life or human health or things like that?
Yes, I love that question. So the first problems that we're tackling are how do you engineer cells to make materials? And not just make materials, but do that in a predictable way where it's fast and you can get to commercial scale in a way that's economically feasible. But essentially, if you look under the hood, what our models are doing, they're learning how cells work in an environment. And there are lots of problems in which that's important. For example, microbiomes. A famous one is the microbiome in our gut that helps our immune system, helps us break down food. When you start to think about systems, approaches like A-Life can be used in lots of different places.
How has building A-Life gone? Like, what was hard? What made it easy or special?
X really understands what it takes to innovate and how much exploring and failure and time and independence and help is needed when you're doing something that is so audacious and hard and just barely possible. Another thing is some of the infrastructure like X's design kitchen.
And the design kitchen, of course, is our rapid prototyping sort of tech shop on steroids here in the middle of X.
Yes. We talked to the DK and said, hey, design kitchen, we wish we could do this really hard thing. We wish we could sample little bits of liquid very fast and then instantly freeze that liquid with liquid nitrogen. And we need to do that because cellular metabolism moves fast. We're trying to capture things that happens on the seconds time frame. They just said, okay. And they built us a prototype and now we're collecting that data.
20 years from now, A-Life has profoundly changed the world. Like, what's happened? How does the world look different?
In the future, manufacturing looks like local biomanufacturing factories with a lower carbon footprint and totally new materials that even stretch our definition of what a material is today. For example, living medicines or self-healing bricks. We've barely scratched the surface and there is huge potential.
Well, thank you for doing this with me. I'm so excited for you, Relly. I'm so excited for the A-Life team and to see what you guys go do to make a dent in the universe.
Thank you.
I really enjoyed our conversation today with Brad and with Relly. Humanity is starting to move from biology as a science to biology as an engineering discipline. If we can find ways to use technology to help nature help us, then as humanity's problems come at us faster and faster, we're going to be able to answer those problems faster and faster. That's why I'm so excited about Heritable and about A-Life. Next time on the Moonshot Podcast, we're going to talk about some exciting and totally off-the-wall ways to make and store energy.
No one had ever built anything like that before. I was so excited by the prospect of changing everything in the wind business that I really wanted to try and see if I could make it work.
We thought, hey, this is the thing worth doing, but if we're going to do it, we got to do it now.
This is the Moonshot Podcast, a peak action production for X, the Moonshot Factory, with me, Astro Teller. You can find more episodes wherever you get your podcasts and on our YouTube channel. If you want to take your own moonshots, we've developed a masterclass certificate course. Check it out in the show notes below. If you enjoyed this podcast, don't forget to subscribe. It helps other people find out about this series.